Hello guys, it is my pleasure to present here about our lab training module on the photostabilizing strategies for the single molecule fluorescence microscopy. I'm Andy In from Cosa Lab from McGill University. So today we are first going to talk about some introduction about the single molecule fluorescence microscopy. It is a powerful tool for investigating biological entities. It can help us to obtain the dynamic information of many biological processes, such as the interactions between proteins and DNA. It can also allow us for the observation of objects with smaller size, like bacteria, subcellular structures, and virus, which cannot be studied by conventional fluorescence microscopy. One of the main challenges in the single molecule imaging is that the fluorophores have very strict requirements. A single fluorescent molecule must be detected and distinguished among billions to trillions of other solvent or host molecules. For the fluorescent signal not to be obscured, a high brightness is required, which means the fluorophore must have a high absorption coefficient and quantum yield. It also needs to have good stability to withstand light irradiation. Here, for example, a typical trace with very big fluctuation is hard to interpret. So we really need the photostability for long-time monitoring. However, the group probes alone they are not good enough to meet the strict requirements for the single molecule fluorescence. So our strategy here is to quench the triplet excited state. In a typical experiment, the ground state is excited to the singlet state and then relaxes back to the ground state by emitting a photon. Ideally, this cycle would be infinite. However, from time to time, the molecule can go through intersystem crossing to the triplet state, which is critical to blinking and photobleaching. Our strategy here is to use the reducing agent first to form the triplet germinating radical empire, which after a very fast escape form the free radicals and go back to the ground state with the help of an oxidative agent. We can also use other triplet sequencers to facilitate the intersystem crossing to form the singlet germinated radical and then go back to the ground state by back electron transfer. There are many triplet sequencers that are well established in our lab, like Trolox, M-propyl-gallium, ascorbic acid, and beta-mercaptoethanol. There are many techniques for the super resolution imaging, and here the total internal reflection fluorescence is our choice. It is an easy single molecule microscopy technique and commonly used in imaging cells. And it allows for the selective excitation of surface-bound fluorophores, and non-bound fluorophores are not excited. And so it will result a very low background and makes it easier to identify and analyze the immobilized sample. For example, here it has a better signal-to-noise ratio and resolution compared to the normal epifluorescence microscopy. And now I'm gonna show you how to test this triplet state quenchers with the single molecule experiment. We first make a cover slip. We first grab a piece of glass slip, which was cleaned by perennial solution and acetone before. And then we grab a piece of silicone spacer, which is used to create the chamber for our single molecule experiment. And we dry them all with nitrogen gas. Then we put the silicon spacer in the middle of the glass slide and then inject PEP solution on it. Now we put our slide into the oven and inject the PEC solution and incubate for some time. While we are waiting for the incubation, let's briefly talk about the structure of our slide. On the right is the structure of our slide. So the glass slide was covered by a PEC layer, which was attached to the biotin on the other end. We have a DNA strand, which on the one end is our fluorophore, the other end is the streptavidin. And with the interaction between streptavidin with the biotin, we can bound our fluorophore on the cover slip. The triplet state quenchers are floated in the chamber, and the performance of the fluorophores are tested using TIRF. Okay, now our cover slip is covered by a packed layer and ready for the next step. Now we put two silicon powders on top of the silicon spacer, and so our chamber is now completed. And then we are gonna inject the buffer with our fluorophore in the chamber and image them in the single molecule microscopy. We are gonna image the fluorophores alone without any triplet state quenchers. And then we are gonna image the fluorophore together with the triplet state quenchers and see how the performance improves. Of course, there are many other experimental details, but I'm not going through them today. But typically speaking, without any triplet state quenchers, the fluorophores will die in several seconds. But as you can see here, with the help of the triplet state quenchers, the fluorophores last much longer. Each of the bright spot is one fluorophore. And with the help of some triplet state quenchers, they can last as long as 100 times longer than without triplet state quenchers. Thank you very much for watching our video today. Please subscribe our channel and see you next time.